This is part B, not 2, but B, of the lecture Cell Structure and a Little History for InBio 104, Biology of Plagues. In the previous part of this lecture, we were talking about the basic structure of prokaryotic cells, and we were specifically focusing on an aspect of their cells called the cell envelope. We talked about capsules, for example, which are part of the cell envelope, and we talked about extracellular or externally facing structures like fimbriae and pili. Now let's talk about the cell wall. This is a particularly important topic for anybody interested in medical microbiology because there's a lot of medical relevance to the cell wall. A lot of antibiotics, such as penicillin, kill bacteria by destroying the cell wall. That should tell you that the cell wall is really essential to prokaryotic cells. And it's essential because it protects the cell from water stress, also known as osmotic pressure or stress. In other words, it prevents cells from bursting when they're in environments where the saltiness on the inside of the cell is not the same as the saltiness on the outside of the cell, which causes water to move via osmosis, creating internal pressure and lysis. In order to protect bacterial cells and archaeal cells from such a fate, they have a cell wall. The location of the cell wall is immediately outside the cell or cytoplasmic membrane. Its composition is typically that of most of them are made out of very rigid layers of carbohydrate and amino acid based macromolecules. These are structures that we will talk about in a future class. In the domain bacteria, one of our major groups of prokaryotes, the cell wall is typically made out of a very large type of macromolecule called peptidoglycan. Okay? So peptidoglycan is essentially a chemical structure made by mixing carbohydrates and amino acids, sugars and protein components. It's incredibly strong, and it's found only in the bacteria and only in the cell walls. We will use a process in class called gram staining, which I'll talk about in a moment, and the gram stain is used to determine how exactly the cell wall of a bacterium is organized. It's a stain procedure that is done in labs across the world, in medical labs, in hospitals and research facilities, and it helps us to identify bacteria and to choose what antibiotics are best for killing it. In the bacteria, there are 3 groups with regards to cell walls. There are gram positives, gram negatives, and what we call gram intermediates, also known as acid-fast bacteria because they can be stained using an acidic staining protocol. The gram stain only works on gram positives and gram negatives. The cell wall organization determines whether a bacterium is gram positive, gram negative, or gram intermediate. On the left-hand side of this figure are gram positive cell wall structures, and on the right are gram negatives. Gram positive cell walls are characterized by a very thick layer of peptidoglycan, shown here. This is in direct contrast to the very thin layer of peptidoglycan in a gram negative, shown here. This structure here is the cytoplasmic membrane. Notice that it is internal to the cell wall on both these bacteria. So this is inside the cell, this side, and so is this, inside. Okay? So as I said in the slide before, the cell wall structure is external to the cytoplasmic or cellular membrane. In the gram-positive bacteria, all they really have is a thick layer of peptidoglycan. In the gram-negative bacteria, they're a little bit more complex. They're actually much more complex. So gram-negative bacteria have this really thin layer of peptidoglycan, and they have a lot of space, okay, here and here. It's a space called the paraplasmic space, okay, and it holds a fluid called paraplasm. which contains a lot of digestive enzymes. Just outside of that, this structure right here, this whole structure, okay, is called the outer membrane. The outer membrane has a couple of other names too. It's also called the lipopolysaccharide, I'm running out of space, aride layer, or LPS. Okay? And in medical microbiology, it's really important to note that the LPS, or outer membrane, is often very toxic. So if you get infected with a gram-negative pathogen, like certain kinds of E. coli, Escherichia coli, and you get a fever as a result, that's your immune system reacting negatively to the outer membrane of that gram-negative bacterium. It's reacting to these little sugar structures that are part of that external outer membrane. So that's what the cross-section looks like of a gram-positive and a gram-negative bacterium. In the case of gram-intermediate bacteria, okay, the most commonly known gram-intermediate bacteria are the Mycobacterium. For example, Mycobacterium tuberculosis. It's the species name for the pathogen that causes TB or tuberculosis. And it has a cell wall made not out of peptidoglycan, but out of a structure called mycolic acid. More in that on class later. Now this is a stain you're going to get some hands-on experience with. It's called the Gram stain. The Gram stain is useful in gram-positive and gram-negative bacteria. It's a stain that differentiates between the two. Okay? In other words, it'll allow us to tell whether something is gram-positive or gram-negative or gram-intermediate. So the Gram stain always has a capital, or should always have a capital G in it, because it's named after Christian Gram, a Danish microbiologist who invented this staining procedure, and it's something we're still using today, even though it actually originated in the 1800s. There's more on that in your lab handout. In your lab procedure, you'll learn how to do this protocol, so I'll only spend a moment here, but the point that I want to make is that gram-positive bacteria will stain purplish blue, and gram-negative bacteria will stain pinkish red. Gram-intermediate bacteria will stain a mixture of these two colors, so you'll look at them and some will look pinkish and some will look purplish, and you really won't be able to see a consistent pattern no matter how carefully you do the staining. Some won't stain at all. So this is only useful for gram-positives and gram-negatives, but that's an awful lot of pathogens, so it's a great stain to use for medical research and other purposes. All right, we're just going to think briefly now about the insides of a prokaryotic cell. We'll be looking into some of these inner aspects in more detail as we go forward in the case. Once you're truly inside the cell, you're in the jelly-like space, or the jelly-filled space, called the cytoplasmic region. The cytoplasm is a thick jellyish fluid. And in there, you're going to find a variety of nutrients and salts and water. Okay, that's what's in cytoplasm. Now, floating around in that mixture are some very important structures. One, of course, is the genetic information for the cell, which is the chromosome. Okay, so in most bacterial cells, there's one circular chromosome. Okay, just one. Now there are exceptions to that, and an exception that's really medically important is Vibrio cholerae, a gram-negative pathogen that causes cholera. It has two circular chromosomes, and they're not identical. Okay, they're not a pair, they're just two chromosomes. Now also in terms of genetic information in cells, many bacteria contain plasmids. Okay, more on those later in the class, but plasmids are small extra chromosomal molecules of DNA that can carry helpful information for bacterial cells. Now, also in cells are structures that are called ribosomes. The ribosomes are just shown as dots in this picture here. Okay, they are structures that are used by the cell to convert RNA into protein. So they're protein-making structures, and they're found in all cells. There are also a variety, there's a spelling error there, variety of inclusions or particles inside cells. None are shown here, but you should know they exist. For example, they can store up energy reserves in the form of starch. And that's what we're going to talk about. That's all we're going to talk about in this lecture with regards to the basics of a prokaryotic cell. Next, we will briefly consider how eukaryotic cells are formed. So most of the things that I just spoke of a moment ago in reference to prokaryotic cells are listed in the central part of this slide. So here we see on the left a prokaryotic cell. It's a different cartoon, but it's the same basic design. And on the right, we have a eukaryotic cell. This is probably an animal cell because I'm not noticing a cell wall, but it could also be it could be a protozoa, although they also have cell walls. This is probably an animal cell. So let's think about what's similar here. We still have a plasma membrane, and it is bounding the cell, and it is indicated in blue on this figure. We still have that watery fluid referred to as cytoplasm filling up the inside of the cell. We still have DNA, except in the case of the eukaryotic cell, the DNA is in linear chromosomes, not a circle, but a line, and they occur in pairs. Okay? Something called being diploid. And all of the DNA is in the nucleus. Remember, U means true and carry means nucleus. And this purple structure here is the nucleus. Okay? That dark bit at the center of it is a particular region of the nucleus that we don't really need to get into in this case. Okay? So the nucleus is bounded by membranes, another feature or hallmark of eukaryotic cells, and the nucleus contains the chromosomes for the cell. We also have ribosomes, and the ribosomes in eukaryotic cells are found embedded in a layer of membrane called endoplasmic reticulum. It's radiating out around here, and it's continuing through here and here. Okay, so that's where the ER is. And it is holding many ribosomes, or holding the structures responsible for protein synthesis. It also contains a lot of proteins, and it plays a role in transport in the cell. Ribosomes are also found just distributed through the cytoplasm in eukaryotic cells, as is the case with prokaryotic cells. Now, this cartoon implies that eukaryotic cells are typically bigger. We used to say that was true, but in the last 10 years we've learned that in the ocean there are many incredibly tiny eukaryotic cells, so we can't really make that generalization anymore, and so I won't really support that statement here. So using the same three categories that we applied to our discussion of prokaryotic cells, let's think about the basics of a eukaryotic cell a little bit more. Let's think about appendages. So do eukaryotic cells have these extracellular appendages? And the answer to that is that some do. So some have flagella. For example, spermatozoa use flagella in order to locomote up the reproductive tract of females. It's part of their function. They have a reproductive function. Their flagella are structured quite differently from bacterial flagella, but they serve a similar purpose. Some animal cells also have extracellular structures. For example, structures called cilia that are found on the epithelial or surface cells lining our reproductive and respiratory tracts. When women ovulate, it is cilia lining epithelial cells in the reproductive tract that move the egg down from the ovaries lower into the reproductive tract. Whenever you breathe, you're using respiratory cells or epithelia that are lining your respiratory system, and there are cilia that need to move deep in your respiratory system, and they're moving mucus up and out of your lungs. And in fact, it's the impairment of those cilia from cigarette smoke that is one of the reasons that smokers are much more susceptible to respiratory infections. So they do have extracellular appendages, and they are made out of protein, and they play typically a role in motility or movement. Do they have a cell envelope? Yes. Most of them have cell walls. The animal cells don't, but the algae, the fungi, the protozoa, and the plants all have cell walls. And do they have a plasma membrane? Yes, they do have a plasma membrane, and it's made very similarly to that found in a bacterium. Eukaryotic cells don't have capsules, however. That's a bacterial cell feature. Do they have a cytoplasmic region? Yes, they do, and in that cytoplasmic region, you have a watery fluid full of salts, sugars, proteins, and other small molecules. You also have chromosomes. They aren't found free-floating in the cytoplasm. They're found encased in a nucleus. You do have ribosomes. They are found in the membranes of the endoplasmic reticulum or free in the cytoplasm. And you also have additional structures that are known as organelles that are mainly structures wrapped in membrane. And an example of an organelle, it's very, very important, is the mitochondria. That's plural, it's mitochondrion for singular. It is an organelle that is responsible for ATP synthesis. Okay, something that the plasma membrane does in bacteria, but in eukaryotes it happens at the mitochondria. Okay, so let's try to sum up like similarities and differences, and this cartoon depicts one way of thinking about that. The center of this Venn diagram talks about similarities. So in all cells we have DNA as the genetic material. I would argue that it's in the form of chromosomes as well, or I would add to that. In all cells we have ribosomes responsible for protein synthesis, and we have similar basic metabolism. In other words, we have a way of making and using ATP, and those ways are pretty similar from one cell to the next. Okay? Other similarities I would say would be the cell membrane, okay, and the cytoplasm. Okay, all cells have those things. Something that prokaryotes have, they always contain or almost always contain cell walls. They're single-celled, and they don't have membrane-bound organelles. In other words, they don't have a nucleus, they don't have mitochondria. Eukaryotes do have a nucleus, so we could say they contain a nuclear envelope. That's the membrane for a nucleus. They often contain cell walls, so plants plus fungi plus algae plus protozoa. Okay. They include multicellular organisms, although they also include many microbes. And in general, they have a more complex and extensive arrangement of DNA. They have something called chromatin. So how have we learned so much about cells, and when did we begin this learning? In order to study cells, we had to have microscopes. As I said before, they're very, very tiny, and you can't study them without being able to see them. And in order to see them, you require a microscope. So who invented the microscope? So there are two answers to that, two scientists that were alive concurrently and doing their work concurrently. So I'll mention them both, although one is not really more important than the other. So the first I'll mention is a Dutch merchant. His name was Anton van Leeuwenhoek. And he lived in the Netherlands, or what's today's Netherlands, but formerly Holland. He was a merchant and a naturalist or amateur scientist. And in the era, or in the years 1673 to 1723, he wrote a series of letters to a society called the Royal Society of London. That society is still in existence today. I think it's the oldest natural science group still in existence today. And in those letters, Van Leeuwenhoek described small or tiny creatures that he referred to as animalcules. and he included a number of drawings and descriptions of them. He had examined samples of his own feces, scrapings of teeth, pond water, anything he could think of, he looked at it. And the way that he looked at it is that he devised a really sophisticated magnifying glass, and a reproduction of it is shown there on the left. The lens is in that little tiny area at the top, and he would put his sample on the head of essentially a pin, make adjustments, and study. And he probably devised these initially to help him look at merchandise such as fabric or material that he was shipping, so that he could do things like count the thread count in fabric, and then he thought of applying those same magnifying structures to the world around him. So that is documented as one of the earliest magnifying tools, but it wasn't a true compound microscope. That was invented by another fellow. And that other fellow's name was Robert Hooke. He was an English scientist and surveyor, sort of a Renaissance man of many trades. He was also a fellow in the Royal Society of London, so he was aware of van Leeuwenhoek's work, and he devised the first compound microscope. Okay, a compound microscope is one where you have both the objective and the eyepiece, and they are located at opposite ends of an adjustable tube. So you were working with compound microscopes in lab recently. You were using with binocular ones that magnify using each eye, and Hooke first came out with just the single tube style that you can still find in many high schools today. So just like van Leeuwenhoek, Hooke looked at a whole variety of samples, and when he was looking at a cork sample, type of wood sample, he noticed these regular patterns, and he drew them, and they're shown in this figure here, which was published in a book called Micrographia, very, very famous book. So the cork is worth mentioning because based on his investigations of cork, he's the one that came up, or is credited with coming up, the term cell. Okay? And that was the beginning of what we call cell theory, or the idea or the truth that all living things are composed of cells. So he wrote about this in Micrographia, and also in Micrographia he documented many microbes. For example, a fungus commonly found on bread. And so he may be the first person to officially document microbes, or it may have been van Leeuwenhoek. The record's not entirely clear on who came first. One thing that clouds that view is that Robert Hooke was very proficient in English, whereas van Leeuwenhoek spoke Dutch and was uncomfortable in English and a bit reclusive, so Hooke's work was better disseminated or shared with others. So with that, I will conclude this lecture, and I will see you in class soon.